Welcome back. This is Dr. Vishal Trivedi from Department of Bioscience and Bioengineering, IIT Guwahati, and let's continue our discussion about the sexual reproduction in flowering plant. So, what we have discussed in our previous lecture that we have actually understand the importance of sexual reproduction in the plant and how it was evolved over the course of different groups within the plant, like how it was evolved from algae to the angiosperms. So, angiosperms are the only plants which are which have developed the flowers, and they have the well developed the sex organs within the flowers. So, in the previous lectures we have discussed about the structure of the flower, and within the structure of the flower what we have discussed that every flower has 4 parts. One is called the calyx, number 2 corolla, number 3 androecium or male sex organ, and number 4 the gynoecium or the female sex organ. So, now we are going to start the discussion about the male as well as the female sex organs, and within the male sex organs we have the structure known as the stamen. So, as you can see in the given slide, the structure of a stamen. So, stamen are modified leaves and they are involved with the production of microspores or the pollen grains. Each stamen is consist of a cylindrical massive or thread like filament and a broad terminal bilobed anther. So, each lobe of an anther has 2 pollen sac. So, depending on the number of the lobes present in any anther, the anther can be classified either as the dithecous or the monothecous. In the dithecous anther, the 2 lobes are present. So, they have the 2 lobes, one on this side and the other one is on this side, and each lobe is further divided into the 2 pollen sac. But when these kind of anthers are matured, these 2 pollen sacs are going to be fused with each other to give you the 2 pollen sac which are filled with the pollen grains. Whereas, in the case of monothecous anther, 1 lobe is present and the 2 pollen sacs are fused. So, one lobe actually contains 2 pollen sac in the case of monothecous anther, but at the level but when it is getting matured, the 2 pollen sacs are fused to give you the single chamber. So, in the case of dithecous anther, there are 2 chambers filled with the pollen sac, whereas in the case of monothecous anther, there are only 1 chamber which is filled with the pollen grains. So, development of the male sex organ or the anther part is having the multiple steps. What are these steps? So, in one step you have to first develop this cavity where the pollen grains are going to be evolved or developed, and in the step 2. So, that step 1 is called as the development of pollen sac or the pocket where the pollen grains are going to be developed and stored, whereas in the other step the the cells which are present in the pollen sac are going to go for the further division, and then they will evolve to generate the pollen grains. So, let us see how the development of pollen sac or the or the microsporangia, development of microsporangia is taking place in the angiosperm plants. So, this is the structure of a pollen sac which is under the course of development. What you will see that the development of pollen sac begins with the differentiation of archaeosporial cells in the hypodermal region, and it happens on all the four corners of the pollen sac. So, these are the four corners on which the pollen sac is going to be developed. So, because of these cells, these cells actually goes for the periclinical division, periclinal division. So, these cells actually go for the periclinal division to give rise a subepidermal primary parietal cells and a inner primary sporangious layer. The layers of primary parietal cells divide by successive periclinical and the anticlinical division to form the concentric layers of the cells, concentric layers of the pollen sac. The wall layer from the periphery, to center has multiple cells. So, the outer cells which you can see here is called as the endothecium, and it is a single layer of the cells, and outside to the endothecium you have the single layer of epidermis. The layer of endothecium possesses the fibrous thickening and they remain thin walled and constitute stromium, and then below to the endothecium you have the 1 to 3 layers of the middle layer, and cells of these layer normally disintegrate in mature anther, and then there is a single layer of tapetum. The tapetum cells may be uni, bi or multinucleates, and these tapetum cells are actually further divide. So, the cells of primary sporogenous layer, normally divide to give you the sporogenous tissue. So, sporogenous tissue is actually the tissue which is responsible for production of pollen grains. So, let us see how the pollen grains or the microspores are being formed within the pollen sac. So, microsporangiasis is so during the development of microsporangium, the cells of sporangious tissue, which is actually we have discussed in the previous slide, the required in the pollen sac, may divide in further planes and finally give rise to multiple plane and finally separate from each other to function as the microspore mother cells. So, microspore mother cell is being formed from the sporangious tissue. So, sporangious tissue which is present within the pollen sac divides further into multiple times, and that actually give rise to the microspore mother cells. These microspore mother cells are diploid. So, some of the microspore, so there are multiple microspore mother cells are formed. See, some of the mother cells are being used as a source of nutrition. So, they provide the nutrition to the other microspore mother cells, and these other microspore mother cells actually goes for the active division, and then they are actually the cells which are going to form the spores or the pollen grains. So, there are two classes of microspore mother cells. One, the class of microspore mother cells which are going to be disintegrate, and they will provide the content for the nutrition to the actual microspore mother cells. See, each microspore mother cell has a diploid nucleus, and the each microsporocytes or the microspore mother cell then develops an internal layer of callus which breaks the cytoplasmic content of the prominent diploids, which breaks the cytoplasmic interconnection within the microsporocytes, and these separated microsporocytes. So you can see that there are multiple mother nuclear cells present within the sporangious tissue, and then these mother cells are being separated from each other by a layer of callus, and that actually will give you in single individual microspore mother cells, and then the single individual microspore mother cells then go for the reduction division, because they are the diploid cells. So, they go for the reduction division, and through the process of meiosis then they segregate the nuclear content, and then ultimately what they are going to have is the 4 haploid nucleus, and then within the these 4 haploid nucleus then go for the cytokinesis, and because of that cytokinesis it is going to develop the microspore tetrads, and these microspore tetrads are actually contains the 4 pollen grains which are enclosed within the single cell wall. So, cytokinesis of this microspore mother cell or the divided microspore mother cell results into the 4 separate cells which are arranged in a tetrad, as you can see here. So, the tetrads which are present in the which are being generated from the microspore mother cells can be of different types. In general, you can have the tetrads, which is in general you can have the 2 different types of tetrads, which is tetrahedral or the isobilateral. So, these 2 forms of tetrads are more popular, whereas you have the other way of arrangement. So, how this arrangement happens within the during the development of microspore, because of the plane of cytosol division within the microspore mother cell when it goes for the division. So, as I said, the usual way of getting the microspore tetrad is either tetrahedral or the bilateral, isobilateral, but in some cases you can also get the decussate, linear or T shape. So, these are the alternate microspore tetrads which are also being formed. So, the one question comes is, what is the structure of a microspore or the pollen grains? So, microspore or the pollen grains are actually the haploid and nucleated uninucleated minute spore produced in large number as a result of meiosis in microspore mother cells inside the microsporangia or the pollen sac. Each pollen grain of angiosperm has two cells, one is vegetative cells and the other one is generative cells, and it also has a distal aperture for the germination. So, the pollen grains are generally globose in shape, and through several other shapes are globose in shape, but in some cases they are also oval or to the polyhedral. So, it depends on the particular kind of plant species and the particular kind of so that according to the plant species, some plant species also have the oval or the polyhedral microspores or the pollen grains, but in general they are found globose shape. In a individual pollen grains, it has two layer. It has two major cell walls. One is called the exine, which is outside, and the other one is called the entine, which is inside. And the exine is the outermost layer, and its shape, it is composed of a material which is known as the sporopollentin, and this is actually a organic polymer which is composed of the resistance for the oxidant as well as the other kind of physical damage. Whereas, the inner side is called as the intine, and that intine is the inner layer and it is mainly composed of the pectocellulose. In angiosperm pollen grain, the exine is mainly derived from the tapetal substances during the sporogenesis, and intine is derived from the male gametophyte during the development. The exine at one or the multiple places, the pollen grain has the a very thin exine. So, these are the places where the pollen grain has a very thin exine, and these are the places through which which are being used for used as an aperture for through which the pollen tube emerges at the time of germination on the stigma. See, pollen grains are as I said, pollen grains, and as we discussed, the pollen grain is composed of exine and intine, and both the exine as well as the intine is composed of a large quantity of the organic substances, and because of that the pollen grains of some of the plant species are very good source of nutrition, and they are always being used in the form of the formulations or the tablets, and they provide the nutrition. Whereas, in some cases, because these are the organic substances and pollen grains are always being dispersed in the air, they also been a very good source of allergen. So, they are also causing the allergic reactions in the human being. So, in that way the pollen grains are having the both beneficial effect as well as the harmful effect to the human being. Apart from the once the pollen grain is produced onto the within the pollen sac, the pollens they are also have to be liberated from the pollen sac, and this process of so the changes which are going to happen within the pollen sac, so that the within the pollen sac, so that the pollen grains are going to be released from the anther are called as the dehiscence of anther. So, dehiscence of anther is actually a process which is required, so that the anther becomes fragile and it actually disperses the pollen grains into the environment. So, you can imagine that this is a kind of a pollen grain the anther, and this is the pollen sac. So, one sac and two sacs. So, during this process what happen is the pollen grains which are being stored within these sacs are getting dried up. So, the first process is that the pollen grains are getting dried up and they become more like powdery like appearance. Okay. So, so they become powdery like appearances, and this powdery like appearance within the pollen sac is important because it will help them to disperse in the subsequent step. Then after this the further drying up happens, and the because so in this process what will happen is the partition between the two lobes is going to be broken down, and then ultimately it is going to be a single chamber which is going to have the filled pollen grain. So, the second step is that the further drying will break the partition between pollen sac and that is going to give you the one chamber. This means in one anther, if it is a deciduous anther, you are going to have the two chambers, one by the disintegration of the partition between the two pollen sac and the one by the disintegration of the another pollen sac. So, that is how you are going to have the two chamber. If it is monodeciduous, then you are going to have the single chamber. Now, as the drying continues, that actually will give a pressure on the outer layer of the cell, and the outer layer of cells which are actually called as the endothecum. So, and the endothecum cells so as the further drying up continues, the outer layer of the endothecum cells actually goes for the dehydration, and when they will go for the dehydration, they will contract, and so you can imagine that the endothecum cells are present on the outer layer and they will actually going to contract, and because of their contraction, this chamber is going to shrink or will going to have the inward pressure, and because of that this chamber is going to be broken from the top, and as a result the pollens are going to be dispersed in the environment. So, after that the pollen sac is going to be broken, and that will disperse the pollen grains in the environment. Okay. There are other mechanisms also in the descent of anther as well as the breakdown of the pollen sac to disperse the pollen grain, and but in general this is the only mechanism which has been adopted for most of the plants for doing the descent of the anther, so that the pollen grains are going to be dispersed. Now, once the pollen grains are being dispersed, the pollen grains from the one plant will go to the next to the stigma of the female sex organ, and this process of going from the movement of the pollen grain from the pollen grain from the pollen sac to the stigma of the female sex organ is known as the pollination. So, movement of from anther to stigma is known as pollination. This pollination could happen between the plant 1 to plant 2, or it could happen within the plant. Okay. And because of that, the pollination could be of two nature. One is called self-pollination, which is this one, and the second one is called as the cross pollination. within the self pollination, okay. So, self pollination means the pollination of the travel of the pollen grains from the within the same plant, and that could happen either in the same flower or between the two different flowers. So, the pollination could be of two types. In one case where the plants will be having the bisexual flower. So, in this case the single flower, the anther will go to the stigma of the same flower, and the number 2 could be where the plants the anther will go from the second flower. So, that will be either that could be either the unisex in the case of unisexual plants or unisexual flower. No, so in that case the movement would be from one flower to another flower of same plant. So, this mode of the movement is called as where the pollen grains are going from the one flower and within the flower is called as the autogamy, whereas in the case of the condition where the pollen grains are growing from the one flower to another flower, but within the same plant, and that is called as the geitonogamy. Now, you can imagine that you have a plant and you have one flower here. Okay. So, if it is within this flower, or if you can have suppose one flower here, okay. So, the pollens probably can travel and then go to here, and if it is like this, then this is called as the geitonogamy, whereas if it is like this, then it is called as the autogamy. So, within the self, as you can see on the PowerPoint slide, I have described the self pollination. So, in this case what you will see is that there are two conditions, one where the pollen grains are pollen grains will come from the same flower and then they will go to the stigma of the same flower, and this mode of the self pollination is known as the autogamy. Whereas, in the other case the from the same plant the pollens will come from the one flower and they will go to the next flower, and that kind of self pollination is known as the geitonogamy. So, both of these modes have the further subdivision or further classifications. So, within the autogamy we have three multiple conditions. So, the first condition is known as the cleistogamy, second is known as the homogamy, and third condition and these are the specific circumstances in which these things are happening, is called as the bud pollination. So, what is mean by the cleistogamy? The cleistogamy means that in these kind of plants they will follow the self pollination because the flowers never open in these kind of cases. So, the cleistogamy means the flower never opens. So, once the flower is not going to open, then they have no option but to follow the self pollination, and examples of these classes are oxalis, okay. Whereas, in the case of the homogamy, the anther and stigma matures at the same time. So, I am sure we will discuss this, but you know that the self pollination, the success of self pollination and further fertilization is required that the anther as well as the stigma should be mature, means the male sex organ or the female sex organ should be matured at the same time. Otherwise, what will happen is that if the anther is matured and female organ is not matured, there will be incompatibility, and because of that, even if there will be a self pollination, there will be no further fertilization. So, in these cases, because the anther as well as the stigma matures at the same time, it favors the self pollination, and the example in these cases are mirabilis. Now, the third condition is called as the bud pollination. So, you know that once the flower is formed, it is enclosed in the form of bud. So, as soon as it is in the form of bud, it actually remains immature or it is not fully mature. But in these kind of specific cases, the anther as well as the stigma is matured while within the bud only. So, because the anther and stigma are matured at the same time within the bud itself, that actually favors and you know the bud is closed. So, it actually brings the anther and the stigma very close, and that is how it will actually facilitate the self pollination. And the example in this class is most of the cereals like wheat, rice, etc. Okay. So, this is about the self pollination. There are other mode of self pollination, and that is called as the geitonogamy. And geitonogamy we have already discussed. Geitonogamy means that the flower 1 is doing the pollination of flower 2, but of the same plant. Okay. So, now what the question comes is, what is the advantage or disadvantages of self pollinations? So, there are several advantages and there are several disadvantages of the plant following the self pollination. What are these advantages? So, what are the advantages of self pollination? Number one and the most important advantage of going for the self pollination is that it increases the chances of pollination, means it increases the chances of pollination. And you know that if there are more chances of pollination, this means there are more chances of the fertilization. This means there are more chances of having the offsprings. This means, and that offspring means the more productivity, because more number of embryos and more number of seed is going to be formed in these kind of plants. And that is why if you have seen that in one of the bud pollination, the most of the cereals as well as the most of the food grains are present, like wheat, rice and all those, because they are actually going through this kind of cell pollination, and that is how the efficiency of their production is very high. Number 2, because you can go with the self pollination, you are going to get the anther or the pollen grain from the same plant, and because of that the purity of a particular species will maintained throughout the reproduction process. So, the purity of the species will be maintained. Number 3, because the self pollination is happening within the single plant, it does not require to produce the large quantities of pollen grains. So, it does not require to produce large numbers of pollen grains. Number 4, because the self pollination does not require any external agent for facilitating the pollination, it also can conserve the energy by not to produce very brightly colored or very beautiful flowers. And so, in that case also it does not have to produce flower with colored petals or scent or the nectars, because these are the things, means colored petals or the scent or the nectars or any kind of other features. These are the features which are required actually for the attracting the agents. Okay. But in these cases there is no agent involved in doing the pollination. So, because of that the plant can conserve a lot of energy in producing the color or producing the nectar. Since they have so many advantages, the self pollination also has many number of or several type of disadvantages. What are the disadvantages of self pollination? Number 1 and the foremost advantages or disadvantages of the self pollination is, and that anyway we have discussed, that one of the most important advantage of the self pollination is that it maintains the purity of the particular species, means it does not allow the mixing of the genetic content of the other plant, and that is actually advantages when it goes for the self pollination, but that itself brings the disadvantage, because if you are actually doing the propagation of from the plant 1, you cannot have any method to acquire the qualities. So you can imagine that if the plant 1 has few qualities and plant 2 has few qualities, but if the plant 1 is going through the self pollination and plant 2 itself is going through the self pollination, then these two species will remain pure, but they will not be able to have their cross breed. So, that actually will not going to give them the plant 3. Suppose they go for another kind of mode of cross pollination or not for self pollination, then they might produce a plant 3, and that actually is going to have the Q and Q2 both. This means they are going to have the qualities of plant 1 and they are going to have the qualities of plant 2. But since both of these plants are going for the self pollination, they will not going to have the other qualities. So, in that cases the offsprings which are going to come from the plant 1 will be very weak in terms of acquiring the additional features. So, the offsprings are going to be weak. Means when I say weak, means they are not going to have the additional features which are present with the other plant, which means these offsprings are not going to have the variation in their qualities. And that is very, very important. As soon as if you remember, we have discussed, when we discussed about the sexual reproduction, we said that sexual reproduction is being evolved in angiosperm mainly because the sexual reproduction is going to bring the lot of changes in a particular species, and those changes may be good or may be bad. And other point is, as the plant 1 is going through the similar kind of genetic material, it is actually getting weaker and weaker over the course of generation as it goes from one generation to many generation. Number 2, if it goes on self pollination, there will be no species or new species of this same plant are not going to be produced. So, these are the two disadvantages of the self pollination. Means the self pollination restrict the plant to its own family, and that's actually is going to be a problem because it does not allow it to acquire the traits from the other person. So, what is the problem? So, what is the solution? So, solution of this problem is that if this plant will go for the pollination with the neighboring plant, and that kind of mode of pollination is called as the cross pollination. So, in the cross pollination, there are several factors which are actually contributing towards the cross pollination. So, what are the factors which are why we are discussing about these factors? Because you can easily ask, if the plant have the choices to go for the self pollination or the cross pollination, why it will go for the cross pollination? Because cross pollination means it actually being dependent on some agent to carry the pollen grains. Okay. So, Even then, there are several factors which actually does not allow the plants to go for the self pollination, and in those cases plants have no choices but to go for the cross pollination. What are these factors? Number one factor is that the is called as the hercogamy. So, there are many plants which are actually having the mechanical barriers to protect their stigma or their anthers, and in those cases these kind of plants cannot get the self pollination because they have the mechanical barrier. This mechanical barrier has to be removed by an external agent to facilitate the pollination, and in these cases the cross pollination is only possible. So, in these cases we have two types of mechanical barrier. One barrier, so we have, so in the hercogamy means that the plants are having the mechanical barriers and that protects the self pollination. So, that does not allow to have the self pollination, because you can imagine that you have a flower like this, but this area, so you can imagine that you have the stigma, you have the anthers on the side, right, but this middle portion, which is actually the stigma, is covered with a membranous sheet. So, with this kind of if it covered with a membranous sheet, and this kind of membranous sheet is known as the, so the membranous sheet which covers the stigma is known as the whereas, so this gymnostegium is actually a membranous sheet which covers the stigma of these plants. So, if the gymnostegium is going to cover the stigma, it will not facilitate the entry of the pollen grain into the stigma, and it will not allow the fertilization. Similarly, in some cases a membranous sheet is also covering the pollen grains. So, these are the pollen grains. So, in these cases these kind of sheets are called as pollinia, and the pollinia is also doing the similar kind of function, that it protects the pollen grains to be released spontaneously to facilitate the self pollination. Thank you.